Good evening, everybody. I'm Amrit Mudda. I'm Professor of Neurosciences, and I'm also the admissions tutor for all the undergraduate degree programs in biological sciences at the University of Southampton. Thank you very much for joining us for this very exciting webinar today, which is the first of the webinar series that we're going to offer for the next few months. This webinar is really designed to enhance your understanding of what scientists do, biomedical scientists do, biologists, zoologists do, what kind of research questions they're interested in asking, what kind of research they do. And also it's an example of the kind of university lecturer that you would be taught by if you were to do a degree program at the University of Southampton, because we are very strong believers in research-led teaching, and all of our lecturers are active research scientists, and so their teaching is very much delivered with a flavour of the research that they do. And we thought that the best way to demonstrate this is to host this lecture series, and I'm very pleased to invite you to this first lecture series, which is going to be delivered by Dr. Nicole Pryor, who's a lecturer in stem cell biology. Before I hand over to Nicole, I just want to tell you a little bit about what this next hour will look like. Nicole will give her presentation on the research that she does. After that, there will be an opportunity for you to ask questions. The best way to do that is for you to type your questions into the chat, which is on one of the columns on your screen. On my screen, it's on the right-hand side of my screen. Type your questions in there. Please do that even as Nicole is speaking, because I'll be monitoring all these questions, and I'll make sure that each one of your questions is asked either to Nicole or to myself at the end. Your questions can relate to anything that Nicole presents about her research, or your questions can be related to any other aspect of biological research, or the life of a scientist, or how she or I decided to do what we do, or what it's like to be a student in Southampton. We can give you links to current students whom you can contact through what we call a UniBuddy system, or if you have other questions about what it means to study biological sciences at university, or anything in particular about our biological sciences programs, then please do ask those questions, and we will endeavour to answer all of them before the end of the presentation. The final thing to say is that this webinar is being recorded, so if you feel, don't worry if you feel like you're missing anything, because you'll be sent the recordings afterwards. If there are some questions that you have in mind, but somebody else has asked those questions, please use the icons that are available in the chats to like the questions, because that means that we're aware where there are some questions that many of you have in mind, so we'll elaborate on those more than others. Okay, without further ado, I'm going to hand over now to Nicole Pryor, who will first and foremost introduce herself and give you her background. It's very important for you as young, aspiring, young students, some of you who may go on to be aspiring scientists, to understand what the journey of current scientists has been. How did they get to where they've got to? And Nicole will start off by giving you a little bit about her background and how she came to do what she's doing now. Nicole, over to you.
Thanks, Amrit. So hi everyone, thank you so much for joining today. As Amrit said, I just want to tell you a little bit about how I got to where I am now. So quite a few years ago now, I did my A-levels, and I did A-levels in biology, in chemistry and maths. And then from that, I always was quite drawn to biology as a subject at school. So I did my bachelor's degree, and that was in biological sciences, focusing on molecular genetics. And I had the opportunity actually to do a year in industry during my bachelor's. So I spent a year working at AstraZeneca. And from that year, in this sort of working environment in the lab, I realized that research was something that I really enjoyed and I wanted to go further in. So I decided to do a PhD. So I moved actually to Germany, and I did my PhD in Germany, which was a great opportunity, and it's where I was first really introduced to developmental biology. I worked on embryos there. And following my PhD, I then moved to the University of Cambridge here back in the UK, and that's where I started researching the liver and working with organoids, which we will discuss a little bit in the coming lecture. And then a couple of years ago, I moved to the University of Southampton. I was appointed a lecturer in stem cell biology, and I also started my lab. So as you can maybe tell from this slide, my lab, we focus on liver biology. And here what you can see is a mouse embryo. The liver is highlighted here in red. And this is actually at day 10. This is the mouse liver from the embryo 24 hours later, and this is 48 hours later. So already you can get an idea of how quickly the liver is growing. So there's lots of questions to answer about how it can coordinate these processes and how this is done. And this is what we work on. So in today's talk, I'm really going to talk to you about biology and zoology from a developmental biology perspective. It's a chance to give you a bit of a taster lecture, and then we'll discuss a little bit more about the research we do in my lab. So first of all, why study biology and zoology? Well, it really gives you the chance to explore physiology and behavior of organisms from across the breadth of the animal kingdom. Even get insights from extinct animals because we have the fossil record, and lots of different types of animals. So we have, for example, more basic animals such as the sea urchin and the jellyfish. We have amphibians, we have birds, we have insects, and of course we have mammals and primates. And the insights we get from this may be at the level of intracellular molecules, so individual molecules within the cell, or all the way up to the study of whole ecosystems. And using these insights, we can develop our understanding of evolution. We can understand how animals behave, and this can help us develop strategies for conservation. We can look into more molecular techniques like neuroscience and how the brain works. And of course, what we're going to discuss today, embryonic development. So how do these animals actually form from that single fertilized egg? So idea about, as a lecturer, what I teach on. Here I teach on the second year cell biology module, on the second year vertebrate development module, and the third year evolution and development module. And of course, I also have fourth year project students working in my lab. So I just want to take a minute to really explain how fascinating developmental biology is. So of course, we're going to go from this single fertilized egg to a whole functional organism with many different cell types, different specialist organs. And what's incredible is this has to occur through a coordinated interaction of the cells that are actually going to make up the body. And what I mean by that, it's a very different process. If we think, for example, here we have all the component parts of a car. So to make the car, we put the parts together. But if we think about making an animal, these parts don't exist yet. What we have is this single fertilized egg. So the component parts actually have to make themselves as they develop. And this is why it's so fascinating. So really, there are two main questions that we're asking when we're thinking about developmental biology. We're asking, how does this fertilized egg give rise to the adult body? And secondly, how does this adult body give rise to yet another body? How can it reproduce? And by looking into these questions, we can get insights into other things. So for example, how can understanding the process by which these embryos develop give insights into evolutionary processes? So let's think about that a little bit here. Now, this isn't a new question. You can see here, these are illustrations actually that were made by this man here, Ernst Haeckel, and you can see they're from the 1800s. But what he was doing was he made illustrations of different animals, because they're actually labeled in German, but bear with me. So we have a fish, a salamander, a turtle, a chicken, a pig, a cow, a rabbit, and at the end a human. But he looked at these different animals and illustrated what the embryos look like at early stages of embryonic development all the way to later stages. And what I think you can nicely see through these illustrations is actually very early on in the embryonic development, there's a lot of similarity in the structures. For example, they all have these folds, which are called branchial arches, and they all have a tail. And what this tells us is the common ancestor of these various species, the embryo of that common ancestor would have had these features. Now, of course, as evolutionary time passed, these various structures in the embryo of that common ancestor will have specialized in different ways. So, for example, the branchial arches of the common ancestor in the fish lineage turn into, whereas the branchial arches in the human lineage will have turned into structures in our face and in our jaw and our neck. And also, of course, you may already know this, but as the human embryo develops, a sort of tail actually forms and then it regresses. So by looking at our embryonic development, we get insights into our evolutionary past. And this leads me on to the next point, which is vestigial structures. Now, these are part of an organism, but they no longer perform the main function. So if we think about ourselves, for example, some of you, for example, may be able to wiggle your ears. And this will be a remnant of the ear muscles that our ancestors had, which would have allowed them to turn their ears towards the direction that sounds came from. You may also notice when you get stressed, sometimes you get goosebumps. Our ancestors would have had to raise their body hair in order to appear larger to scare off potential predators. So what we see are these evolutionary vestigial structures, and they give us clues into the life of these common ancestors. Now here we have two different salamanders. We have this cave-dwelling salamander, which lives in the dark, and we have this surface salamander. And you'll notice two things, I think. One, the salamander that lives in the dark has much less pigmentation. And secondly, if you look where the eye should be, you'll see that it's also blind. Now, what's interesting, if we look at this from a developmental biology perspective, is if we look at the embryo of this cave-dwelling salamander, actually, the initial eye development is normal in the embryo. But as development progresses, the growth of the structure slows down, some of the cells die, and this results in some under the skin. So what that tells us is actually in the evolutionary past of this species, of this cave-dwelling salamander, its ancestor would have lived in the light and had functional eyes, but it's lost this over time. And another really nice example of this comes from the spotted dolphin. So here we can see various dolphin embryos progressing through different developmental stages. And we're going to look at the formation of the blowhole. Now, the blowhole, of course, is located at the top of the head in the center. But interestingly, it develops from these two nasal pits that are initially either side of the head structure. Over time, they migrate, they move towards the midline of the head. They then fuse and slowly move up the head structure. So rather than simply creating a top of the head that turns into the blowhole, what we actually see is it is formed from two separate nasal pits that come together, fuse, and then moves into the position we would expect. So what we have here are vestigial dolphin nostrils. And another nice example from the dolphin is we have these embryos here, you can see progressing through development. And if you look here, you'll see that actually these dolphins during their embryonic development have hind limbs that start to grow and then they slowly disappear. So it would seem strange that they would go through this effort to form a hind limb when it's just going to disappear. But this is the vestigial dolphin hind limb. So this is important because using these two observations, we can see the separate nostrils merge and move to the front of the face to the top of the head, and the hind limb ancestor of the dolphin likely had a nose or snout structure more reminiscent of a land animal, and that they also would have had hind limbs. So that's the inference we can make from just looking at the modern embryos of dolphins. We can also test this hypothesis by looking at evidence from other areas. So we can look at the genes that are involved in forming, for example, the hind limb in these dolphin embryos, and we see that actually it's the same set of genes that are used, for example, in other mammals, so in the mouse, for example. So this would suggest that this is an evolutionary vestigial mechanism from a common ancestor. And finally, if we are theorizing that the dolphin has evolved from some sort of tetrapod land animal with a nose, with a snout, well, we can also look into the fossil record and ask, do you see any evidence for this? And actually, yes, we do. So what we see actually is a creature known as a basilosaurid whale, which existed about 34 to 40 million years ago, which looked a little bit like this. So it had hind limbs and it had this snout structure. So you can see that even looking at modern day embryos, we can get insights into the evolutionary past of how these various species have developed. Now, generally speaking, when we're doing developmental biology, we use these 7 core model organisms. And you can see we actually have a plant here, we have various invertebrates, we have vertebrates, we also have the mouse here as a mammal. And you might ask, well, what's the point in that? You know, maybe you're more interested in human biology, maybe you're interested in medical sort of applications of biology and research. But actually, we can get a lot using these various model organisms. So for example, of course, here we have a human, but you can see we have a digestive tract, so does the fly. We have a nervous system, so does the fly. Our body is organized into these segments, which we're going to discuss in a moment, but so is the flies. We have circulation, skeletal structures, so does the fly. So we can actually learn a lot from analyzing these model organisms. And in fact, 75% of human disease genes have a counterpart in the fruit fly. And even we can use even more simple organisms. For example, 50% of the genes in yeast can actually be replaced by human genes, and that yeast will survive. So there's a lot of conservation. So the same genes are used in lots of different species. So let's talk about the main crucial factors. Vertebrates come in many different shapes and sizes, yet the key developmental stages are observed in all of them. So we go from a fertilized egg. So here in the case of the zebrafish, this cell here is the egg. It's on top of a yolk sac. What happens first is known as cleavage. So this cell divides many, many times. The volume doesn't increase, just the number of cells. These cells then form what's known as a blastula. And this is where we get sort of a ball of cells, which then start to move around each other during gastrulation. And this allows for the formation of different types of tissue. So this will then allow for neurulation, which is where we start to form the head structures and the brain, and also then segmentation. So when the body becomes segmented, and this is where we form, for example, our backbone and our muscle. In the case of the fish, as I say, the zygote, the cleavage, blastula, gastrule, and segmentation is common to all vertebrates. And here you can just see in this movie, this again is a zebrafish embryo. So what you see here is the cleavage stage. You can see it's the same volume, but many different cells. Then we start to have this movement down around the yolk sac. So this is now a blastula. And there's lots of cell movements occurring here. Again, as we were saying before, this all has to be coordinated. We're starting to see the formation of tissues now, so during gastrulation. And what we're going to see is the head structure will be on this side and the tail will elongate in this direction. And you'll start to see segmentation of the embryo. And the segments you'll see come up as these arrow-shaped blocks of muscle. So here now you can see the head, and we can see here we've got the body, and hopefully what you'll see in a second is you can see these arrow-like segments coming up. There you go, going from this side all the way down to the tail tip. And what I hope you can appreciate from that is the multiple changes, cell differentiations, events that had to happen all within, so this is in hours, so all within 27 hours. So it's all happening very quickly. It needs to be highly, highly regulated. Now, we were saying that here we can see segmentation, and we've already discussed that actually across many different species, a lot of the same genes are used. As a way to illustrate that, I just want to show you segmentation in these various arthropods. So arthropods are invertebrates with an exoskeleton, but what we can see are in the various segments of the embryo of an insect,